Yeah, thank you all for coming. I'm sure we'll have some more people trickle in here in a little bit. But I wanted to introduce Camila Rueda from Colombia. She's been here for almost 6 weeks on an internship and also helping us to train our volunteers in the flasking lab and the DNA lab area, which is just fantastic because it's really helped us get that moving along a little bit faster than we would have. I've known her for 2 years now, I think so.
Yeah, it's been...
this is her second lecture here at Selby Gardens. She came in early 2024 and gave a talk on her research at the time, which is on mangroves. And then we met up and spent more time at the botanical conference in Cartagena in Colombia, also in 2024. Just finishing her master's degree.
Right on.
You're going to go back and
graduate.
Yeah.
Bureaucracy at universities, but I finished like a couple months ago.
And for undergraduate, she had a double minor, international studies, and in biodiversity studies and environmental services. A lot of experience for such a young person working in the field. And I like the way that she describes herself in one of the, like on ResearchGate, botanist interested in conservation. That says it all. That's great. So,
thank you. Well, thanks for the introduction, Bruce. I appreciate everything that I've done so far. It's been an amazing 6 weeks at Selby. I've had the best time also, not only at Selby, but in Sarasota. It's been such a wonderful city to be in. So before I start, I've seen you've been looking at it, this wonderful landscape. I won't be talking about this ecosystem today, maybe in a future lecture. But this is the Páramo. This is about 3,200 feet above, meters above sea level, so 3,200 meters above sea level. This is an ecosystem that's pretty vulnerable to climate change, which is something we'll talk about a little bit later. And these are the Espeletias, also called frailejones or frays in English. And these are also plants that are pretty much endangered, but bring big service to all Colombians because they are the ones who help filter out the water, all the moisture in the mountains. These mountains tend to be really dry, but they have a lot of cloud and moisture seeping in. So this and the mosses and also other plants work to get the water to stay in the mountains. So here I will be talking today about catering to plants' needs, but we must also consider the other perspective where plants also cater to our needs. So first up, this is a heat map where if we make a timeline from here to there, we have 1970 over here and 2020 at the end. This is how much, on average, our temperature has changed in the world. So when we see the blue lines, it means the average temperature went a little bit down. Gray line, no change. And what do we see as we get closer and closer to the present? The world is warming up. Our average temperature is getting higher, and that poses challenges to the equilibrium that we have in the Holocene, which is one where humans have thrived. We as a species have lived for almost 100,000 to 100,000 years, but it hasn't been until the stabilization of weather since 10,000 years ago where agriculture became and agriculture will all civilization. Another thing to consider in this lower part is biodiversity. This is the standard biodiversity in 1970, and we can see as this changes from green to gray, it means extinction. So this is how many. This is the change in species that have gone extinct each year since 1970 till 2020.
Wow. We
can see it increases with each year, unfortunately. Now, 45% of plants are threatened with extinction. So that's almost half. So about 1 in 2 species of plants are threatened with extinction right now. It might be due to habitat change, to climate change, to overpoaching, and overall land transformation made by humans. So, how can we protect plant biodiversity in specific in a changing world? Now, there's various actions that can be done to preserve plants. I'm going to go a little bit over them. We have governmental action. So this is big, widespread, multilateral agreements that a lot of where countries and whole continents can compromise on plant conservation. We have a bunch of different institutions such as the UN and everything that spreads out from it, like the Convention on Biological Diversity, which in turn made a couple decades ago the IUCN, which provides us with the red list as a reference of which species are endangered, and governments take action on what's on this red list. We have multilateral agreements such as the COP, which this year was in Colombia, where there were a ton of biodiversity compromises made by countries. And in a more specific way, we also have conservation efforts that go all the way into law. So this is not, we will try to protect, no. CITES is something that's very, very specific and that's very ingrained in law. For example, all of us who have worked with orchids know that we need to abide by these measures in order to either work with them or purchase them. Now, on a bit more specific side, there's in situ conservation. So once we already have some guidelines as to what to do, we can take actions in those places where we want to preserve biodiversity. How can this be seen? There's protected areas, biodiversity reserves. There's different ways in which we can prioritize biodiversity in restoration, because there are many restoration projects ongoing right now where they might say, hey, we planted 10,000 trees, but what if those 10,000 trees were the same species? We're not recovering forests, we're not recovering ecosystem services. So prioritizing biodiversity in restoration practices is very important to guarantee that whatever we're restoring will have a future. Other things to consider in a changing world is assisted migration. The world is changing at a rate. The world has always changed in temperature and weather, and of course, plants and animals have responded accordingly to it. However, right now it's changing at a pace that's faster for them to respond. So sometimes we might need to consider if species tend to migrate towards a mountain upslope. Maybe if we're trying to restore an upslope mountain part, maybe we might need to include species that might benefit from that area. And a little bit more specific too is maintaining reproductive success. If we're restoring biodiversity in certain areas, we need to make sure that if we plant those trees, they have the pollinators available to produce more seeds and to keep and to keep growing the place, because if we don't guarantee this, we're just planting, everything will eventually die and we would have lost our time. And more specifically, ex situ conservation, which is our role as botanical gardens. Here we have various strategies that we can use where we are the safekeepers of biodiversity within our own institutions. Some example is the Kew Millennium Seed Bank, where they not only do research on biodiversity, but they're also safekeeping seeds for the future. And that seed bank is made up of a lot of biodiversity, which is different from the seed bank that we know that's in Norway. Those mainly have crop related seeds, so peas, beans, but this one has native trees. They even have orchid seeds stored in there, so it's a different focus. and a pretty important one. Another example of this is the Million Orchid Project at Fairchild, where they're growing their orchids at a great rate. So mainly, not only that, but living collections here at Selby, we're known for bromeliad, for our amplified collection, and that's really important also to safekeep those plants, because when a big storm comes along or if some unfortunate thing happens, we will have some genetic reservoir within us to help aid and restore those populations. So after all I've said, what role does someone like me, a botanist, have in all of this? And just a spoiler, it's a little bit of everything. So this list, you can see, what role should I play within the great scheme of plant conservation? I'm not a policymaker, I'm a botanist. So what can I do? First of all, I need to make sure that whatever we're doing, whichever plant we're trying to save, is taxonomically accurate. So we need to make sure the plant is what it is and not some other one. Check out the distribution of those plants and their phenology. So when are they flowering? When are they fruiting? And for that, it's also very important the partnership with local communities, where we can also keep someone that's out in the field keeping an eye on the plants and seeing how they're doing. Afterwards, we might want to do field work to take specimens and seed collections to bring out into our botanical gardens, start with propagation procedures, and think about different ways in which we can do in situ and ex situ conservation. So if we go out and do a botanical expedition and we figure, oh, this area has more species than this other area, this might be more open to prioritization than this other one. So that will be into in situ. And for ex situ, that's where we will keep the living collection, for example, in place in a botanical garden or a seed bank. And after all, we're not a policymaker, but we can give out policy recommendations. For example, the ones who do the IUCN Red List assessments are us, experts on our fields. I'm going to focus more on ex situ conservation from a botanical garden's perspective. And I'll give out two main examples of things I've done throughout my career, mainly in collaboration with Universidad de los Andes in Colombia and the Cartagena Botanical Garden. First up, mangroves. As I told you before, I'm going to talk a little bit about mangroves and the different ways we can protect them in an ex situ collection. So a little bit of context, this is within the Colombian Caribbean mangroves. We have the same species you have over here. So we have the black mangrove, Avicennia, the white mangrove, Laguncularia, the red one, Rhizophora mangle, and the buttonwood mangrove, Conocarpus erectus. All four of these, you can find them along the path over here, right next to Selby. However, there's one that's not here in Florida. It's Feliciera bentami. This is a recently, kind of recently described species. And it's the tea mangrove. And there was this, they thought it was only one species all along the Pacific and Caribbean coast. But then they realized the one that's on the Caribbean, in the Colombian Caribbean coast, is different. And that's why it's recently described. And if the original one that they thought was already endangered, this one now with reduced distribution was even more endangered. And it is. I did the IUCN assessment, and it is. So what information do we have about this plant? Well, once we figured out the distribution, talked with local communities, and we're lucky enough that at the time I was working in the Cartagena Botanical Garden back in 2021, it was the second half of the year, so they were fruiting. I went out to the field looked out for them. And I want you to see something very particular over here. It's something that happens with mangroves all the time. You might see a fruit, but once it opens, you see a very big seed and already plant growing there. So it's not like your average bean seed where you don't see any leaves or anything. This is already growing. This is already metabolically active. This is practically, in plant terms, a toddler. And that gives us a little bit of a problem if we want to work through our ex situ strategies. So there's definitely no possibility of seed storage. And, well, there's also a few populations that were known in the Colombian Caribbean. So this is where our journey starts. As I told you before, this was a species, they thought that this was all one species, all spread throughout Colombia and Panama. But they realized that this, well, it's a new species. And that's represented by these black dots. That's in this little area in the Panamanian Pacific, not in Colombia, just in Panama. And over here in the Colombian Caribbean, so very few known populations. I've seen some reports that it's now over here, but hasn't been updated yet. So once we figure out, well, this is a newly described species, what do? We go out and do our taxonomy work, where we go out and take some samples, make sure they're passed out into the herbarium, and we always check with references to make sure that that's the right species. And once we were sure it was Pelicera benthami, we got our hands working. I worked with local communities doing different workshops. Some of them were not aware that this was a different mangrove. Others indeed were aware, because they might live near the mangrove, but this species is very scattered, so it's not like really common to see it all around. It's very hard to find, and so we worked with local youth communities that were interested in environmental issues. We gave out talks, we went out to the field with them, we had a bunch of fun, and we collected a bunch of seeds with them. The next part of our process was determining propagation protocols. So I tried different ways of growing this mangrove. I used different light treatments, different substrate treatments, and finally settled on shadow and sand, and that's what worked best for this mangrove. You can see over here on the span of 8 weeks just how much it can grow. It's beautiful. This is where they store all their food, so you can see it's pretty full and it dries up as it grows, so it has a ton of nutrients to grow. Keep that in mind when I show you the orchids next up. And afterwards we determined which was the best way to propagate them. Next year, so 2022, we established a nursery and we were able to grow them and propagate them. And eventually, we opened up an ex situ living collection of mangroves in the Cartagena Botanical Garden. Mind you, the mangroves tend to be at 0 meters above sea level, and this garden spends about 150 meters above sea level. So we're at a tiny slope right next to Cartagena. But even still, the mangroves thrived. Pelicera is doing pretty well, and it even flowered a year ago. So that's like a good indicator that the plant is happy over there. So we have a genetic reservoir just in case from the area where we took them, which is slightly threatened by locals because they are building some hotels in some areas so people can go out on the beach And the other side of where we find this, it's not locals, but it's people from other parts of the country that are also building their hotels. And as they're building their hotels over there, the locals also want their own. So it's a social issue where there are permits being granted to people who are not even from the area to be there. And that generates a backlash response from the community where they're always being told not to ever harm the mangroves, but then they let someone come in and harm the mangroves. So it's something to take into account when we're dealing with this kind of situations, that it's not just, oh, bad people from the community. No, it's a set of different dynamics. It's a dynamic of, well, the people who live in the middle of the mountains in Colombia, and interacting with people from the Caribbean coast, it tends to be a lot of power play. So it's different social dynamics that also come into place when you're when we're talking about environmental issues and the way that the land is being used around it. But if that place tends to have a diminishing population of tea mangroves, we will have that genetic reservoir in our botanical garden. So that's why living collections and exceeded collections are very, very important for long-term genetic diversity. So that's the story for Feliciana and Tammy. And now I'm going to move on to what was my master's, and it's seasonally dry tropical forest orchids. A little bit of a context of this ecosystem. It's that it's an ecosystem that's considered critically endangered. So 50% of what's remaining of this ecosystem is in the neotropics, so mainly all America. And unfortunately, it is very, very fragmented due to anthropogenic pressure. This ecosystem has something marvelous, that as trees are deciduous, so they look at their leaves every year. Due to the dry season, that's why it's seasonally dry, it forms a very thick organic matter in the ground. What that happens is that when it decomposes during the rainy season, it generates just the best soil to grow in. Now you see where I'm going? Agriculture has taken over and absolutely destroyed this ecosystem because it's a very fertile soil. Now, it's mainly threatened by habitat loss, agriculture, urbanization, because if you have agriculture, then you have growth right next to it, city growth. Climate change is also changing the dynamics in which the rainy season is going on. So we might have, if we have rainy seasons that are very intense, we have nutrient runoff. If we have rainy seasons that are not very rainy, then we have massive die-off due to drought. And of course, timber harvest. These trees are wonderful. Their wood, lovely. Unfortunate for them too, because they are very hardy wood. We have some mahogany too between one of these. We have, so unfortunately, there's also targeted loss of species. Now, into our orchids. There's about 81 species reported for Colombia in the tropical dry forest, with 48 genera. But they estimate that there might be as much as 350 estimated species. There's been a lack of botanical expeditions in this area, because some of them overlap with conflict. Because fertile soil, you have a lot of people that want to take over that, and in Colombia we have a land ownership problem, so that makes it harder for some botanical expeditions to go out and see the areas that haven't been affected yet. And I'm talking about 81 species, and it's a pretty high number. However, in Colombia, with more than 4,200 species, the orchids of these ecosystems tend to be overlooked. So that's why I was also trying to focus on this ones, because, well, the Andean orchids have way too much attention for now. So this is just a little wide array of those 81 species, and my main focus was on this Sartopodium paniculatum and Encyclia cordigera. So Encyclia, very beautiful, wonderful, and Sartopodium, which is
Wow.
and it's funny because it's slightly similar to so the seasonality that we feel in the northern Caribbean area is also similar to the one we feel here in Florida. And this is Encyclia cordigera, but there's a cousin of this over here called Encyclia tampensis. And Sartopodium paniculatum also has a cousin over here that's Sartopodium punctatum. So it's funny, we have kind of like similar wide range genus all along the Caribbean area. And they also have them in the Caribbean islands too. Different species, but they are all cousins. Now, once we identify the ecosystem we wanted to work in, the species that we wanted to work in, there comes the collection of the seed pods. So I mainly wanted to focus on ways that we could, germinate these orchids. And just a little bit, we can see that this seed pod is about 10 centimeters. It's about 4 inches long, but look at this, 0.5 millimeters. The seeds. These are tiny. These are known in literature as dust seeds. And Irene, Thassia, and Carrie can confirm they really are super dusty. Like they get blown away by anything super small. Now, what can we do to protect these orchids? First of all, well, some orchid seeds can be stored, but there's more research to be done in this area. Some of them die off pretty quickly after the 6 months in storage. Some of them can last years in storage and still be viable to germinate. But these seeds, as they're so tiny, do you remember the what I told you earlier with the mangrove? That it had this big storage. Orchids do not have that. Orchid seeds are very special in the plant kingdom because they do not have that endosperm, which is the food that they need, that an embryo, this being the embryo, would need to germinate. That's actually all the nutrients. The endosperm is what we mainly eat when we're eating beans, chickpeas, and so on, and that's where all the nutrients from those food come from the endosperm of the seed. Now, these seeds do not have any of this. What do they do out in nature? They have a pretty close relationship with fungi. So fungi will provide all the necessary nutrients for this little tiny seed to grow. And once that seed is already a big plant, its roots will provide refuge for that fungi. And also, once they make photosynthesis too, they will provide sugars to the fungi. So it's a pretty much it's an interaction that's win-win for both of them. Which is pretty interesting. Now that we know this, well, how can we replicate this if I don't have the fungi? That's where in vitro seed propagation comes in handy. For this we use this special type of gelatin that we call agar, and it has all the nutrients that a fungi would provide to the orchid seed in the wild. Now, not only that, but we can also play around with it and add different organic additives. So that would be banana, coconut water, so it will be extra sugars. It will be from the coconut water, a ton of hormones, plant hormones, and that can enhance the growth of the orchids. Now, this will be the goal. This is the ideal. The ideal way to do it. But for this, from the seed to germinate and to finally have enough growth that will produce a flower, it might take from 2 to 3 years. It's a very time-consuming procedure, but it's very important because the way that these plants tend to be propagated is they take off the stalk, separate, and plant it again. So they do it by just separating the stalks, and that's an asexual way of reproducing orchids. You're mainly taking the clones out of it, and that's not good if we're looking forward to having genetic diversity in our collections. So how do we make all this media? A little fun lab class. We mix up everything. We have to adjust the pH. We have sterilization is super important, so we have to keep everything extremely super duper clean to avoid one of the main problems in using this, which is, and I'm sorry if you're eating, contamination. So we might have some bacteria growth, fungi. So fungi can be orchid's best ally or worst enemy, depending on the context. And this is one where it's not good. So in order to avoid all of that, we do a bunch of sterilization procedures all along the course of making this media, and also sowing the seeds. Now, let's assume everything went right. We have no contamination. What can we see? So for Encyclia, these are pictures from my thesis, and I recorded the way that it was growing in the media, and I added different stuff to the media. So this is where I bring in back a little bit the title, we're catering to plants' needs, because I wanted to know, I was asking the plant by doing this experiment, hey, what do you like most? Do you like coconut water? Do you like potato powder or banana powder? And, well, the plant answered. So, the simplest way to explain it is that it didn't grow as much when I was using a combination of potato and coconut water and banana and coconut water. And it loved coconut water over here, where we have a big proportion of plants reaching the final stage of growth, which is this one. So yeah, and we can see like the ways that it's responding to the treatment across different stages of development, and well, that's fun. So now we know it's like, okay, it likes different stuff. It doesn't like when you combine things. Just keep it either normal with coconut water or with potato, which is kind of interesting. We have it over here, but no combination. Interesting. And with Sartipodium, I asked myself the same question. What do you like? Fortunately, it grew in almost all media. In all media, we have different stages, and just to make the connection with fungi, we see that they have this beautiful tiny rhizoids over here. This is where, if it were in nature, the fungi will start to colonize all this. The good fungi, the one that we want with the orchid. But as it is in a very abiotic environment, we don't see that. We just see the way that the plant puts it out, but no fungi colonization. And after asking Sartipodium, what do you like? It also gave me an answer, and it said, I'm picky, I don't like anything. So, just focusing on this one. The ones that mainly reached this stage 6, were the ones that were in the control treatment, which is pretty pronounced. Super picky, didn't like anything. Give them the kids menu, come on. And, these results are very interesting because it paves the way in which we can more efficiently propagate distinct species that might be endangered, that are in endangered ecosystems, in a way where we can improve our practices by knowing what each species wants and being more efficient at what we do. And in the case of Sarcopodium, well, it's even cheaper. We don't need to buy the other stuff. So it's just very, very good to know what plants need, and also how we can use how we can further maximize the use of our resources, limited resources, for conservation. And in Cartagena, at the end of my program, one of the things I'm most proud about was that this used to be a bathroom. And we completely changed it to have an orchid lab where we could do propagation directly. Because the one thing I forgot to mention is all of this was happening in Bogota at 2,800 meters above sea level. So, and these plants were from Cartagena, so completely sea level. And this was happening in very controlled conditions in growth chambers. But we don't want to depend on the institutions and certain areas that are pretty far away to do our conservation work. We want to do our conservation work where the plants are. And that's why we're here in Cartagena. So yeah, we set up this lab with my advisor, Santiago Madriña. And it's a very low cost but very efficient lab, and we've been having pretty good orchid growth so far. And not only that, but part of my work was training Jesus, which is an undergraduate student, and Santiago, which is a researcher at Catering Botanical Garden, into all the different ways in which these species can also be treated, and also doing more and more propagation. This is the final result of my thesis. There are more plants over here, but over 800 individuals were grown from this project and are still growing because it's only been a year, so we need about another year to see if they flower. So they're right now being very well taken care of in the Cartagena Botanical Garden. And all the paths lead me, and the last one, we're not only figuring out how they work in the nursery, but we're also trying to figure out how they can, once being taken out of their flasks, come back again to the trees. So the bigger ones that are doing better, we put them up with Jesus, which you just saw, in different trees across the botanical garden, which is something that's amazing. So in our botanical garden, we are not only doing orchids, but we also have a bunch of native tree species where we can work with. So it's a living laboratory, the one that we can work with. And we're putting up orchids in a place that's their ecosystem and seeing how they do, how they adapt, and if we can finally say that this in vitro has been successful in creating plants that can go back to their ecosystems. Now, well, this is our flasking lab at Selby. So all the paths finally let me here, and it's in its earliest stages. So once again, my project, my master's was 2 years, and it took about 6 to 8 months to get a full-grown plant. So we're mainly starting over here, but it's a good start. At Selby, now we have growing Epidendrum floridense, very, very endangered orchid and important to the state of Florida. Sacoila lanceolata, Sacoila paludicola, which we have been doing a bunch of research since 2018, and Encyclia champensis, a local favorite. And all this work at Selby is the result of 6 weeks of training and of having wonderful conversations and times with Dacia, Irene, and Carrie, which have been wonderful to train and work with. They're even better than my students. They're diligently taking notes. You know how hard it is to get somebody to take notes right now? It's been great. It's been great, and I have a whole team that can take care of the plants. They can keep growing them, and hopefully by Christmas I tell everyone. So you might have some beautiful green gifts.
Wow.
So yeah, that's all for today. Thank you for your attention. Here's my contact if you have any questions. I don't know if anybody. Yeah, over there in the back.
Like what a seasonally dry rainforest is.
Oh yeah, for sure. I will go back a little bit. Yeah, I didn't go into many specifics. So seasonally dry tropical forest is a forest that has seasons. So it's in so as you get closer to the tropics, you don't get fall, summer, winter. You only get rain or no rain. So wet and dry season. And in the seasonally dry tropical forest, you have periods of time where you have almost no rain. And I'm talking like utterly 0 days of rain in a whole month. It might be that extreme. And that fluctuation between very dry and then you have the rainy season, which is crazy, that makes the plants there adapt in different ways. So if we have what's called hydric stress, some plants respond to it in a way that they will lose their leaves, so be dioecious, during the dry season. And then when the wet season comes back in again, they will sprout new leaves. And some others will have different ways of adapting to this very dry moments. But yeah, it's usually from December all the way to April where it's super dry, being the driest months in February and March. July knows too. That's how it is in the Dominican Republic and a lot of Caribbean islands. And then we have the heavy rainy season, which is kind of like the same season here that we have with hurricanes, where September, October, November is heavy rains. And a very light rainy season in June, July. So you get this like super dry, a little bit of rain, a lot of rain, and then the cycle starts again. And that dynamic shapes the way that organisms respond to it. So you have plants and birds and mammals that would respond to all of those changes. And the thing that's interesting is that orchids also respond to that as well, because that's why you don't have as much diversity as you might have in the Andes Mountains, where you always have perfect conditions. But here it's very tough, very tough. So they have a wider distribution, and they also have like different strategies so they can survive those dry months. Because if you're a plant that's in a tree that will lose its leaves in dry season, you're even going to be even more hydric stress than if you're a plant that attaches to a tree that does not lose its leaves. So it's kind of interesting the effect that might have on the plants that live there.
Camila, I'll just point out that Florida is exactly the same.
Yeah.
We were in a seasonally dry, not quite as extreme as in Cartagena in that area, but the pattern's the same throughout the Caribbean.
Yeah, I usually have a picture where you can, aerial picture. You can also see it yourself in Google Maps, where you see Cartagena in March, and it's completely like my hand, like super dry, you don't see anything. And then you look up at a picture from November, which is right at the end of the rainy season, and everything is green. So there's a very, like, even completely landscape change in just a few months every year. Oh, sorry, you had a question.
Yeah, could the mangrove that you've just discovered, is it called the tea mangrove?
It's called tea mangrove.
Could that live in Florida? Or would there be a chance that it would ever be propagated and brought here?
I don't think so, but it might have historically been in Florida, because this, I'm going a little bit into its natural history over here. This mangrove has been around, it's been reported to have been around for almost 45 million years.
Wow.
There's the pollen of this mangrove is pretty unique, of the Pellicera genus, and they found, like, on those big tubes of dirt that they check to see pollinography, where they check to see how the earth has changed, and they've taken out some of those and they've recorded that pollen in areas that are meant to be 45 million years.
Wow.
So that means that it had a big widespread distribution all along the Caribbean, where, well, it wasn't the Caribbean back then, but it was like, geologically speaking, like we were more together. And then the red mangrove comes along like 30 million millions of years ago, and it kind of like overpowers the species. So kind of like, it's also like the dynamic between these two species, where I would say it's kind of hard, and also that one is more sensitive to salt water than the other mangroves I showed you, the black, the white, and the red mangrove. So I think it might not grow in Florida.
We don't want it here.
No, you don't need it here. Yeah.
It would be, if it impacted our native mangrove species, that would be a big problem.
Yeah.
Yes. After you get your carefully raised orchids into the trees in the botanical park, do you fertilize them, or do you let them be on their own, or what do you anticipate will happen with them?
Yeah. So we fertilized them right after they came out of the flasks that they were in, and afterwards it's recommended for them to be watered every day for a month. That's what the recommendation that Fairchild gives. But we were in the middle of the rainy season, so we had a little help with that. And so far they've been doing good, and Ciclia is performing better than Sartopodium in that sense. And yeah, we're trying to still figure out why. That's Jesus' undergrad project that I'm helping him out on, being a co-director with Santiago Madriñan. So we're still trying to get some answers from the plan. We're like, okay, come on, tell us what you think about this change, and then we'll see. But for now, we do water them, but not fertilize them after they're on the trees. Just right after they come out, so they have some nutrients, like the first week or so after they're out of, the perfect, that's like a womb. It's like perfect conditions where they just can keep growing, but you can't keep them in there forever. So we do that, and then, but once they're out of the greenhouse, we don't fertilize them anymore.
Do you think that the additives that you're putting in the agar as part of those experiments, does that inform in any way where these seeds might germinate in nature?
It does. So with Sartopodium, this is not the only species that has been found of this genus that has this type of behavior. It's actually something that could have been expected, because when there's been 2 other studies done in Sartopodium punctatum, and where they added the additives, and it was also super picky. Like, I don't want any of this. I just like my as simple as possible. And what they realized is that, well, these are orchids that tend to grow in very, very dry areas. Brazil actually has the most species of this in Cerrado region, which is super dry, even drier than the seasonally dry tropical forest. So they tend to be under very tough conditions. So they're tough. They're like, I just let me grow without anything. However, Encyclia has a complete opposite distribution as a genus. So we're trying to think about a little bit back. So kind of like when you're at the doctor and they tell you, oh, what conditions that your parents and grandparents have. So we do that the same with the orchids. So we're like, okay, so your cousins and your siblings, like, what do they like? And you can kind of like put the puzzle together over there.
Oh.
And with Encyclia, it goes the other way around, where this is an orchid that its cousins and siblings tend to live along more moist areas with more nutrients around. So that's why it might respond favorably better to having the nutrients in it than Sartopodium. But it's a great question. Yeah, it does inform us a little bit of the life history of both orchids.
Great.
Yeah. Any other questions? Comments? Okay.
Thank you very much.
Thank you very much for coming.
put in a plug for the Cartagena Botanical Garden. It's a beautiful, beautiful place near a beautiful city, and
And we'll have a great orchid collection
Yeah.
that will be blooming soon. We already have an orchid collection over there, so if you want to head out and look at it, but we also have amazing.
so it's been a
trees.
pleasure having you here.
Thank you very
Thank you,
much.
Riz,
Yes,
for everything.
absolutely.
So, thank you for coming. Have a great day.
Right.